Hello, my name is Jacob Bollinger, and today we'll be talking about direct ddPCR refinement for the development of an in-field PCR device by the Fort Lewis College Engineering Department. So PCR stands for polymerase chain reaction, and it is used to replicate DNA to usable levels. So this can be used in the medical field, such as whenever you get a COVID test, they swab your nose and they use that to test for COVID. And the amount of DNA that is detectable at the start has to be amplified to be able to even detect it. So similar with environmental testing, if you have a soil sample or a water sample and you want to test the bacteria that might be living on it, then you have to amplify this DNA up so that you can actually either send it off for sequencing or look at the proteins within it. So the reagents that accomplish this include primers, which these are sequence-specific sets of DNA that the lab creates. And this attaches to a particular part on the target DNA sequence so that you can amplify a specific sequence so you know exactly what kind of organism that you are looking at. So then there's the DNA polymerase, and this enzyme is what attaches to those primers, and it'll build the template DNA, the replicated DNA, with the dNTPs, and then you have your template DNA. So amplified DNA, like I said, can be used for sequencing, and that's to be able to determine what organism that you're looking at. It can be used for concentration analysis and gel electrophoresis, which looks at more of the proteins within the target organism. So there's different methods of PCR. There's real-time PCR or qPCR, and this kind of tells you when your sample starts to be amplified at like what cycle. And then you have droplet digital PCR, which uses the same sort of technology as qPCR, but you partition each reaction into droplets. So what's the point of this? So they're developing an in-field PCR device to help researchers in the field. So water sources can become contaminated with bacteria, and a lot of times they're of fecal origin. So for my research, I focused on E. coli because this is a fecal indicator biomarker, and it is used frequently with surface water contamination to determine where the source is coming from. And so developing methods to be able to decrease the time it takes from sample collection to having your results would benefit public health much more by being able to understand more about the sources and how to prevent them in the future. So the current methods for testing surface water contamination includes taking various different samples along the water source, and then you take them to a lab or you send them to a lab, and there the lab will perform DNA extraction, they'll perform PCR, and then if they're sequencing it, if you're in the case of Fort Lewis College, then you'll have to send it off because we don't have a sequencer. And this can take a lot of time to do, especially the culturing, and that's oftentimes used for concentration analysis. As shown in figure 2, this is what's called streak plating. So basically you spread your sample over this, what's called agar, and this agar is like a smorgasbord for bacteria, and it has every single nutrient they could want, so they grow really fast, but it still takes about 24 hours. So my main goals for this project were to decrease the time from sample collection to getting your results of quantification of bacteria within a sample and identifying those bacteria. And then I wanted to use minimal reagents to increase the affordability and practicality of the in-field PCR device. And also I wanted to make sure that I could develop a direct PCR method, which is whenever you use intact cells instead of DNA extraction. I wanted to develop a method that was reliable. And so there's my main research question. So what is direct ddPCR? So like I said, direct PCR skips DNA extraction. And what DNA extraction is basically whenever you isolate all the parts of the cell, you get rid of them, but you only have the DNA left. And this is used to make sure that the DNA polymerase can build the DNA without basically any interruptions. So I use direct PCR, so you skip that whole step, which can take about 3 to 4 hours sometimes. And then ddPCR, like I said earlier, is PCR that's partitioned into many different droplets. And so here's a quick little rundown of how PCR works. So you have your initiation step, so it's basically just the DNA there. And then you have your denaturation, and this is when, so the reaction is performed in what's called a thermocycler. And so you have different cycles that'll go to different temperatures, and at this certain temperature, the DNA will split for denaturing. And then while it's split, you'll have primer annealing, and your sequence-specific primers will attach to the target sequence where they're supposed to. And then for ddPCR, you have a fluorescent probe. And so this probe is also sequence-specific. And so it'll bind to a specific sequence, and as the DNA polymerase comes along, as shown in this extension step, it'll break apart the probe, and the fluorophore, this green part, whenever it's not next to this negative, the little ball shown in gray, it'll start to fluoresce. And so what's important about this is that whenever you have this fluorescence, you know that it's this specific bacteria, so you eliminate the need for having to sequence. So this is how droplets are generated. On the left is the microfluidic chip design, and right here is where the oil goes in. B is where the PCR mix along with your cells go in. C is the T-junction, which is where droplets are formed, and then D is where the oil and droplets come out. And so this uses water-oil emulsion technology. So basically what it is why water floats on oil. They have different densities, and so you can use that to make droplets. So in figure 5, I have this T-junction shown better. And above over here, you see what's called dripping. That's whenever you're creating droplets. And then below is what's called wetting. And that's whenever, if you have your oil ratio, if you have your oil flow rates going too slow, so what's coming through this channel and this channel, if it's going too slow, you'll have wetting. And if your PCR mix, what's coming through here, is too fast, you'll also have wetting, which can make it a little bit difficult whenever trying to create droplets. So here is a picture of some of the droplets. So here in A and B, these are both the same position on the slide, but A is shown with the overhead light of the microscope on, and the laser as well. But you can see the negative droplet shown here below. And then on B, the overhead light is off, and all you have is the excitation laser on. So you can clearly see the difference in positive droplets versus negative. Similarly, in C and D, you see that C has the overhead light on, and then D does not. So there's been a problem with low fluorescence intensity, and there's two different things that could be causing this. One of it might be droplets merging. And so as shown in figure 8, you can see that the droplets are very uniform, and this is in the collection chamber directly after the T-junction. Another reason that might cause this low fluorescence intensity could be inhibitors that are inhibiting the DNA polymerase from building the replicated DNA. So a little bit more on the merging of droplets. So if you look right here, this is the collection tube. And so it has to travel downward into this PCR tube, and that's what goes directly into the thermocycler. So I think this is the main reason that the droplets have been fusing together. And the reason is because the oil mixture that's used, it's a mixture of oil and surfactant. And surfactant's a very watery-like oil, basically. And as you mix this oil in, it's still very watery, so it turns out to actually be more dense than water. So the PCR mix, which is mainly water almost, that's actually less dense, so that'll float. And so as it's traveling down this tube, as you would see with oil floating on water, these droplets are floating on top of the oil. So what happens is that gravity causes the oil to exit out first, and then you'll see like a distinct line where the droplets are trying to push up against each other to be able to exit the tube. And I think the force caused by this pressure is causing the droplets to push oil away from each other to where they're touching, and then they fuse. So the other thing that might be a cause of this is inhibitors. So what I have here shown in A The green part is the DNA polymerase, and then the purplish blue is the natural substrate that should be used. It's called DNA-C. So that's the natural substrate bound to the enzyme. And this is what allows the enzyme to build the DNA. So in B, we have what's called DNA helicase. And there's two different conformations shown in yellow and then in green, and they're both bound near the active site. And then in C, we have the 30S ribosomal subunit that is bound in two different conformations. And then D, we have two different conformations of the 30S subunit bound to the DNA polymerase. And the issue of this is that if they're bound in the active site, then the substrate cannot be bound to that site. And so the DNA polymerase will not be able to build the DNA. So binding affinity here shown is DNA-C, what was shown in A in the last figure. So this is what's called a negative delta G, and the more negative, the more spontaneous a reaction, which means the more favorable. So basically it just means that you're not having to put energy into the reaction for it to happen. It's actually releasing energy. So BAM-C, which was shown in B, is we see 206, 195, and I put two different conformations of them bound, and then we have the 30S1, so the first two conformations that I had shown in C, and then 30S2, which was shown in D in the last figure. And so what's important about this is that their binding affinities are, yes, they're not as favorable as DNA-C, the normal substrate, but they are still very close, which means it is very reasonable to assume that this would be a reaction that would happen. So conclusions are that through my research, I did discover that this direct ddPCR method did reduce the time to identify bacteria. The droplets uniformly produce within the microfluidic chips, so we know that we can get single cell encapsulation, meaning that you can encapsulate one cell in one droplet so that you know the concentration. So if you have 30 droplets and 10 of them are fluorescing, you have 30% of your sample is contaminated with bacteria. And then we have concentration analysis will also further determine if we need to do PCR refinement. So the future work is obviously we got to fix the droplets from merging, then use the recently completed spectrometer by the engineering department for concentration analysis. It basically just counts fluorescent signals as droplets move by. And then we can assess the concentration analysis reliability, so we can compare that with streak plate cultures and determine if we need to refine. Based on that, we can determine if we need to refine the PCR reagents, maybe look more into the inhibitors, and possibly look into PCR enhancers. Here are my acknowledgements and my references, and thank you.